How?
Liver EphB4 knockout attenuates insulin resistance in mice. To investigate whether EphB4 is necessary for insulin-induced InsR degradation, we designed human EphB4-specific short hairpin RNAs, shRNAs, and conducted stable knockdown of HepG2 cells, extended data figure 7a, b. In biotin-labeled cell surface protein assay, insulin-induced cell surface EnsR reduction, whereas EphB4 knockdown almost completely rescued this effect, figure 5a, b. These results strongly show that EphB4 is critical for insulin-induced hepatic InsR degradation. Figure 5, hepatic EphB4 knockout improved metabolism in mice. A.B. Plasma membrane InsR protein levels were detected in control and EphB4 knockdown HepG2 cells stimulated with or without insulin stimulation by measuring surface biotinylation. Insulin treatment, 100 nm for 10H(N) equals 3 independent experiments. C. Strategy for generating EphB4 LKO mice. WT, wild type. D. E. PAC levels detected upon insulin stimulation in primary hepatocytes from EphB4 LKO mice on an HFD, N equals 5 biologically independent cell samples. F. Gluconeogenesis in primary hepatocytes from EphB4 LKO mice fed in HFD, N equals 4 biologically independent cell samples. G. GTT in EphB4 LKO mice fed in HFD, age, 18 weeks, HFD, 10 weeks, N equals 10 mice. H. Plasma insulin level during the GTT in EphB4 LKO mice fed in HFD, N equals 10 mice. I. ITT of EphB4 LKO mice fed in HFD, N equals 10 mice. J. N. GIR, J. HGP, K. HGP suppression by insulin, L. ISGDR, M. and FFA suppression, N. during a hyperinsulinemia clamp study in EphB4 LKO mice fed in HFD. N equals 6 mice. O. GTT of DB/DB EphB4 LKO mice, age, 10 weeks, N equals 11 mice. P. ITT of DB/DB EphB4 LKO mice, age, 11 weeks, N equals 11 mice. Q. Plasma insulin levels in DB/DB EphB4 LKO mice, N equals 12 mice. For statistical analysis in B and EQ, comparisons were versus related controls, using two-sided students' t-test. P-values in G denoted by asterisks, from left to right P equals 0.037, P equals 0.024, P equals 0.0075, P equals 0.0038, P equals 0.0056. P-values in I denoted by asterisks, from left to right P equals 0.0051, P equals 0.0087, P equals 0.0071, P equals 0.0079, P equals 0.0056. P-values in O denoted by asterisks, from left to right P equals 0.033, P equals 0.018, P equals 0.0063. P-values in P denoted by asterisks, from left to right P equals 0.0060, P equals 0.0016, P equals 0.017. Values are expressed as the mean plus or minus say M. Because total EphB4 knockout mice are embryonically lethal and liver tissue plays an important role in glucose homeostasis, we generated EphB4 liver-specific knockout mice, EphB4 LKO, using the CRISPR/Cas9 technique and the CRELOX-P system, figure 5c. As expected, approximately 70% of the EphB4 mRNA was specifically depleted in the livers of EphB4 LKO mice, extended data figure 7c, and hepatic EphB4 protein levels decreased, accompanied by increased levels of the InsR protein in EphB4 LKO mice, extended data figure 7d, e. Then, we assessed glucose homeostasis and insulin sensitivity in lean chow-fed and HFD-fed EphB4 FL/FL and EphB4 LKO mice. No differences in body weight, glucose tolerance, or insulin tolerance were noted between lean chow-fed mice of different genotypes, extended data fig 7FOH. In contrast, in the context of HFD feeding or obesity, the primary hepatocytes from EphB4 LKO mice showed a greater response to insulin stimulation by increasing PAC levels, indicating that EphB4 deletion in hepatocytes improves hepatic insulin signaling, figure 5d, e. This result was further confirmed by performing a gluconeogenesis assay using these primary hepatocytes, in which insulin exerted a stronger suppressive effect on glucagon activity in hepatocytes from EphB4 LKO mice fed in HFD than in those from control mice, EphB4 FL/FL, figure 5f. In vivo experiments showed that EphB4 mice gained the same amount of weight as wild-type mice, extended data figure 7I, but a marked improvement in glucose and insulin tolerance was observed in EphB4 mice with a reduced basal insulin level compared with the controls, figure 5G. To more accurately quantify in vivo insulin activity and to assess the tissue-specific sites of insulin sensitivity, we performed hyperinsulinemic euglycemic clamp studies. The amount of exogenous glucose required to maintain euglycemia, glucose infusion rate, GIR, during the clamp studies was substantially higher in the EphB4 mice than in the EphB4FL/FL mice, figure 5J. Hepatic glucose production, HGP, and the ability of insulin to suppress HGP, which reflects hepatic insulin sensitivity, were also improved in the EphB4 mice, figure 5KL. Finally, the insulin-stimulated glucose disposal rate, ISGDR, which primarily reflects skeletal muscle insulin sensitivity, and the ability of insulin to suppress plasma-free fatty acid, FFA, levels, which reflects adipose tissue insulin sensitivity, were both increased in the EphB4 mice, figure 5MN. Taken together, these data demonstrate that the deletion of EphB4 in hepatocytes leads to a marked improvement in systemic insulin sensitivity under conditions of diet-induced obesity, and these effects were observed in all three major insulin target tissues, liver, muscle, and adipose tissue. As obesity or insulin resistance is often associated with tissue inflammation 121, we assessed whether hepatic EphB4 deletion altered these processes. Hepatic CCL2 and IL-6 levels were significantly reduced in the EphB4 mice, extended data figure 7J, and the expression of CCL2, IL-6, TFA and the macrophage marker F4/80 was downregulated in the epididymal white adipose tissue, APW80, of EphB4 mice, extended data figure 7K, indicating that EphB4 deletion in the liver results in a less inflammatory state in both the liver and APW80. To evaluate these effects in a genetic diabetic model, we knocked out EphB4 in the livers of DB/DB mice by crossing EphB4 mice with DB/plus mice. Liver weight and the coefficient of liver and WAT in knockout mice were lower than those in control mice, with no change in body weight between groups, extended data fig 8AF. Accordingly, glucose tolerance, insulin tolerance and hyperinsulinemia were all substantially improved, fig 50Q. These results indicate that EphB4 knockdown confers a systemic insulin-sensitive phenotype in mice with genetically induced obesity. Consistent with the findings in EphB4 on an HFD, the hepatic deletion of EphB4 in DB/DB mice significantly reduced inflammation in the livers, extended data figure 8G, indicating that inflammation was ameliorated by knocking down EphB4 in the livers of DB/DB mice. Similar to our findings in the EphB4 stable knockdown HepG2 cells, insulin treatment promoted InsR degradation in a time-dependent manner in the primary hepatocytes from wild-type mice. However, insulin lost its effect on InsR degradation, and PAC levels remained high in the primary hepatocytes from EphB4 mice, extended data fig 8HJ. Thus, EphB4 is necessary for insulin to mediate InsR degradation in the liver.
Pharma